This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, CEIRS. From Microbe TV, this is TWiV. This Week in Virology, a special episode recorded on June 26, 2019. I'm Vincent and you're listening to the podcast all about viruses. Today we're recording in Baltimore, Maryland. That's Baltimore, for those of you who can't understand that. We're at the annual meeting of the Centers of Excellence for Influenza Research and Surveillance, fondly known as CEIRS. Not Sears and Roebuck, CEIRS, because Sears and Roebuck is gone. This is kind of a homecoming for me. I don't know how many of you know this. Flu was my first love. Influenza virus, not flu, influenza virus. I worked with Peter Palese many years ago, but sadly I left for polio. So it's good to be back at a flu meeting. I have a few individuals who are at this meeting to tell you about what CEIRS is all about and some of the research that goes on. And so let me introduce those to you. Right here next to me is the Chief of the Respiratory Diseases Branch at NIAID, Alan Embry. Welcome to TWiV.
Thank you.
Good to have you. And next to him from Icahn School of Medicine at Mount Sinai, Florian Kramer.
Good morning.
We have mentioned your name multiple times on TWiV. I always call you Franz Klammer, the skier.
That's okay.
Yeah. And from the University of Rochester School of Medicine, Jennifer Nyack. Welcome.
Thank you.
And we're going to talk a little bit about CEIRS and the work that goes on here. We have a nice audience. Are you nice? Yes. Do you all work on flu? Who doesn't work on flu? I don't. I'm the only one. That's impressive, actually. I was at a retrovirus meeting recently, and there were about 20 people who didn't work on retroviruses. Anyway, stick around. Are there any TWiV fans here? We have t-shirts. See, I'm going to throw these out at the end. These are World Tour, 2018 World Tour t-shirts. A special design by Sharon Isern, who's a virologist in Florida. And on the back is a list of all the places we went last year, like this. And I will throw them out. But you have to be a fan. If you're not a fan, you can't have it. Let's start by finding out just a little bit about your histories. I want to know how you got here. And Alan, let's start with you. Where are you from?
Sure, thanks. I'm originally from Louisville, Kentucky.
Not much of an accent, do you? Huh?
Yeah, you know, even when I was a kid, people around would ask my father if I was really from Louisville because I didn't say it properly, which is Louisville.
You grew up there, went to high school and so forth?
Went to high school, then moved to the Carolinas where I stayed for a number of years, where I did my PhD before moving up to Washington, D.C.
So what did you do your PhD in?
I was in a mix of biochemistry and cell biology, studying heterotrimeric G protein signaling at Duke University.
Not a bad place.
Oh, pretty good place to be, especially if you like basketball.
Do you like basketball?
Love it, yeah. Well, being from Louisville, Kentucky, it's a basketball state. Then moving to Carolina, that's another basketball state. So yeah, absolutely.
So can you remember when in your past that you got interested in science?
Well, I was interested in science from the word go, honestly, and I haven't thought about this for a while, but I was originally a chemistry major in college, and then incrementally moved toward biology, and actually, I have to admit, it was when I was trying to decide what I wanted to do with my life, I had read Richard Preston's The Hot Zone at one point and decided I want to do that. And then I started learning about what that really meant and decided I didn't really want to wear the spacesuit, but I did want to work with viruses and infectious diseases. So yeah, that's what did it.
The Hot Zone is like mostly fiction.
It is, but back then I didn't know that. It sounded really exciting.
Sounded good. Yeah. So I'm curious about your career. So you're an administrator at NIAID. Right. I presume you don't pipette anymore.
no pipettes. I put the pipettes down many years ago. And probably I should clarify for those who aren't as familiar with the NIH. So there's the intramural side where we have intramural scientists doing the pipetting and research, and what we call the extramural side where I am. And in our branch, we oversee all respiratory diseases. So not only influenza, which is one of our highest priorities, but also MTB, as well as bacterial pathogens. So I began at NIAID in the laboratory of viral diseases many years ago, where I studied aspects of influenza pathogenesis and MHC class 1 antigen processing and presentation. But being so close to the extramural side, I was really enticed by the idea of being able to not only stay really closely involved with the science, but being able to help scientists, and particularly young investigators, succeed in their scientific careers. So I jumped over and began working in basically what I do today. And for many years, I oversaw a portfolio of HIV viral immunology, and eventually became the deputy director of the basic sciences program there, where I oversaw virology, immunology, drug development, epidemiology, and then also got interested in TB vaccines and NIAID's efforts to advance TB vaccines. So when I was given this opportunity to oversee the respiratory diseases branch, I was really excited to be able to do it. And of course, one of our highest priorities is to advance influenza vaccines that are more broadly protective and durable.
Okay, and we'll talk about that and your role in that. Florian, where are you from?
Well, so I grew up in a small village in a rural part of Austria, the south of Austria, about 20 miles from where Arnold Schwarzenegger grew up. I think you can still hear that.
How far from where Peter grew up?
I would say about 200 kilometers, yeah.
Peter Palese, of course, you knew that, yeah. But you prefer Schwarzenegger.
Kind of depends.
And did you spend most of your schooling there? Through high school, I guess?
Yeah. So I was always interested in nature and science, not necessarily the journals in the beginning, more in the real thing. And so, I went to high school there, and then went to Vienna to a small University of Natural Resources and Life Sciences, initially to study food science, because that's what I was interested in. And so they had a undergrad program for food science and biotechnology. And so I got really interested in biotechnology. And so in Europe, in most European countries, you have to do a bachelor's and then a master's before you can do a PhD. So I did a master's in biotechnology, and they required us to do internships at biotech companies. And so I went to Baxter Bioscience at that point. And so they had vaccine production for influenza, including vaccine production in the biosafety level 3+ facility. And so I was working there for 6 months, and I got really interested in influenza. And so I went back and then did my PhD and worked on virus-like particle vaccines for flu. That's kind of what I did in Austria. Okay.
And when did you come to the US?
Well, I graduated in 2010, and then I was looking for good influenza labs because I wanted to go, to a different country. And I had a number of possibilities, and then my wife decided that we were going to New York, to Peter's lab. My wife always makes very good decisions. And so I started a postdoc with Peter, and then Peter at that point was very much interested in stalk-reactive antibodies and universal flu vaccines. And so I got very interested in that too, and that's how my career in the US started.
And you've been at Mount Sinai ever since, right?
Yes.
How many years has it been?
Well, now it's 9 years.
9, all right. Okay, we'll come back to you. Jennifer, where are you from?
Originally, I'm from Buffalo, New York. So.
Okay. Not far from where you are now, right?
No, actually, spent a lot of time in the region, mostly for family reasons, but I actually really like Rochester. There's a lot of different opportunities, different things to do when you consider that it's a smaller city. So
you're used to snow from Buffalo?
Yes.
Rich Condit, one of my co-hosts, spent many years at SUNY Buff was it SUNY? Some university in Buffalo. And then he moved to Florida to get away from the snow. So you went to high school in Buffalo?
Yep. And actually, when I went to college, I wasn't totally sure. I knew I wanted to go into medicine, but I wasn't sure really what path I wanted to take. And ended up taking a virology course, and that's really how I became interested in viruses and the interaction with the immune system. I was sitting in the library studying, I had Field's Virology open in front of me, humongous book, and my now husband came up to me, had no idea who he was at the time, and I can be on the antisocial side occasionally, I suppose. He's like, so what are you doing? I'm like, well, I'm learning about 101 different things that can kill you. He left very quickly after that. But yeah, so I became interested in viruses and host response at that point, and then thought about doing an MD PhD, but at the time I was involved in research, and my research mentor was an MD, and I was, I do regret the decision somewhat not to pursue both degrees, but I was looking at him, he had an MD, he was involved in research, and I'm like, I can do research and still just have the MD. So I went to medical school, finished residency, and had to decide, okay, now what am I going to do with my life? And at that point, it seemed a natural fit to go into pediatric infectious diseases, which is what I did. The fellowship for Peds ID is a 3-year fellowship, of which 2 years are, in theory, exclusively research. So I was looking around for what I wanted to work in, and ended up actually meeting with Andrea Sant, who was involved in the first CEIRS. This was just at the inception of the CEIRS network, and became fascinated with influenza. I've always been interested in pandemics. I'd seen a few kids in the hospital who had severe influenza disease, and decided this was what I wanted to work on. So in some ways, I kind of grew up in the CEIRS network, so to say. So
we'll talk about your research. Do you still see pediatric patients?
Yeah, I have about 25% of my time is spent clinically, and right now, and about 75% of the time in research.
Is that what you originally envisioned as an MD PhD?
Well, when I decided not to do the PhD, I wasn't even sure I was going to go back into research. But once I started fellowship, I kind of became hooked and really wanted to stay in research if I could. So I would honestly like to have a little bit more time devoted to research. Sometimes I feel like I'm just being pulled in too many different directions. But yeah, during fellowship, I started looking at pandemic influenza vaccines, and we made the observation that there was really what was driving CD4 T cell reactivity was memory cells. There was very little response to the unique peptides within the HA protein when you look at CD4 T cell reactivity. And that got me interested in kind of this whole idea of immune exposure history and how that shapes the response, which led to me starting a pediatric influenza study, which is really right now looking at how immune responses develop in children is really my main area of focus.
Do you remember who taught you virology? And was it Buffalo, right?
No, that was actually in Rochester. So.
Do you remember who it was?
It was a whole host of people, so no.
Okay. And the other thing, this guy who asked you, what are you doing? And you said, I'm figuring out. You said he left, but he came back, right?
He came back, yeah.
He's your husband now, right?
He is.
Okay, got it.
I guess I didn't scare him away too much.
No, apparently not. No. Not even viruses can do that. All right. Let's go back to you, Alan. Let's talk a little bit about CEIRS. And remember, we have a pretty broad audience of listeners who may not know about CEIRS, and I'd like them to understand what it is. So what is CEIRS?
So CEIRS is is NIAID's flagship program for influenza. It's a network of investigators, virologists, epidemiologists, immunologists, studying different aspects of influenza, including the basic virology, how influenza moves throughout animal species, throughout the world, how it changes, evolves, the immunology that we as humans the immune responses we need to have to be protected from influenza, and basically understanding many of the important factors that we may need to consider in developing new and improved influenza vaccines and therapeutics.
So whose idea was this?
Oh gosh, it's a little bit before my time in this position, but it started back in 2004 following a blue ribbon panel on influenza, and from what I know, there was a recommendation to establish such a network. So it has been in existence since that time, and from our view, it's been an outstanding success to be able to build this really interactive network of scientists that also helps, I think, bring up outstanding young investigators and train them to be collaborating in the flu field. So
the title is Centers. So how many centers are there?
Right now there are 5 centers. Thank you very much. There are currently 5 centers, but each of those centers, has nodes at other institutions and throughout the world.
Okay, so 5 centers. And you can help tell him if he doesn't remember. Where are they? Where are they?
So we have the Emory, Rochester, Mount Sinai, St. Jude, and Hopkins. Thank you. Sorry, on the spot. I know this. It's just
okay. We have help. It's collaborative, right?
Yeah.
That's the thing here. Okay, and then there are nodes. I won't ask you to name all the nodes.
Please don't.
Okay. Just give me one node. Do you know?
Well, in Hong Kong we have plenty of surveillance research that goes on. What I should say is that throughout the world, in New Zealand or Hong Kong.
So the centers have to be in the U.S., or they could also be.
They don't have to be in the U.S. They're currently in the U.S. Yeah.
All right. And does this exist for any other virus? This kind of distributed centers idea?
I'd have to think about it. The HIV field has the Centers for AIDS Research, the CEIRS.
CEIRS, yeah.
Their mission is a little bit different, where they're more centers to help have cores that facilitate research rather than doing the actual research.
Understood.
Yeah.
All right. And now, when the program started, did you immediately have all of these 5 centers, or did they slowly come online one by one?
I think, again, I don't quite know the full history. Many of them have been here throughout time, but I think there have been some that have changed throughout.
Okay. So can you have new centers come online or get rid of old ones? Is it dynamic, or is it forever?
No. So the current iteration is 7 years.
Okay.
And so if you were to go to NIAID's website, we recently presented at our council a concept that will potentially move forward. There's never a guarantee until it actually happens, for a renewal of these centers.
All right. So you have
another
7 years. Yeah. All right. So you have to renew. So you have to submit
right,
a
and
renewal.
so each of these centers will recompete. If they so choose, and so then we may have the same set of centers, or we could have some new ones as well.
All right. So you may not want to answer this, but I'll ask you. So what's the total budget for everything?
For all things NIAID, or for all things flu, or for all things
CEIRS?
So because it's a contract, we typically don't talk about contract dollars specifically.
Okay.
So I'll refrain from answering that one.
But it's a lot.
It's a significant investment, yeah.
All right. So how does this work? Do you tell them what to do, or do they come up with their own proposals?
No, absolutely. They come up with their own proposals. Although it's a contract, it's what we call a BAA. And so they are leading the science. The science happens at each of the centers and is reviewed, but then also, on an annual basis, they are deciding which directions to prioritize and move forward with. That's, based on the state of the science at the time.
And that gets reviewed in what kind of mechanisms? It's not the typical study section, I would suppose.
Well, so when the original contracts come in, it's reviewed by a study section. But then annually, each of the centers has a scientific advisory board of external scientists that comes in and talks to them about how their research is going and where they might want to potentially consider moving more or less effort.
All right, so they make a proposal, and it has to fit in line with your vision of what CEIRS should be doing?
Well, yeah, I mean, I would say, generally speaking, we have a broad outline of where, the kinds of areas we would like CEIRS to be working in, but ultimately, it is up to them and, based on their scientific expertise and where they see the priorities to determine exactly the direction where they're going.
And so you're flexible in accepting what the actual investigators are doing in the field, right?
Yeah, absolutely. It's basically, investigator-driven science, but within certain, we want to be, supporting the highest quality and highest priority science to be able to get us to, better understanding the virus, as well as being able to inform, new vaccines, new interventions.
So there's a very nice website, NIAIDSEIRS.org, and it talks a little bit about what CEIRS is. And one big component is obviously research.
Sure.
Right? And it's not all about universal flu vaccines, right?
No, absolutely not. That's definitely true.
So besides, that's what we're going to talk a bit about today, but besides that, what other kinds of research do you support?
With the CEIRS, you mean?
Yeah.
So the CEIRS, I would say, I think that they inform universal influenza vaccine research, but we're not looking to them to be necessarily designing and developing the next universal vaccine. I think, again, they've been very instrumental at understanding a lot of the basic virology. There are cohorts within the CEIRS that are used to understand, responses to influenza vaccines, the immunology that you see, immune responses you see following vaccination or protection or infection that I think are very important. And really also understanding one really important feature of this CEIRS is that they are fairly unique in their ability to understand the human-animal interface, which is so very critical for being able to prevent potential pandemics and being able to predict which viruses may be emerging that we need to be paying specific attention to.
Mm-hmm. And another component is surveillance, right?
Right.
And that involves not only the centers but what you called the what did you call them? Nodes.
Yeah,
I'm trying to remember. Cancer nodes.
Yeah, absolutely. So there are surveillance, surveillance across countries throughout the world, animal surveillance that investigators are actively looking to see in animals and in some mammalian species, especially swine, which viruses are circulating, and then what are the particular features about those viruses that may make them a potential threat to humans.
So the data from all the surveillance, is that publicly available?
Yes, actually it is. So there is a very well-oiled process where all of the surveillance data and sequences go through our data coordinating center, and then those are fed into publicly available databases, yes.
Mm-hmm. Okay. There's also on the website, creation of research tools.
Sure.
What is that about?
So the CEIRS investigators have freely available reagents and resources for investigators who are interested in doing flu research, maybe those that are already involved in the field or those wishing to get into the field. And so that can be anything from viruses, from cells, to reagents for doing immune flow cytometry in animal models. So there are antibodies as well as a number of other reagents.
Mm-hmm. Yeah, I saw a list on the website of what's available as well. Do you have a communication to the public component of the CEIRS centers?
So it's something that I think that the CEIRS are actually, I think, doing a good job of trying to communicate to the public about the research that they're doing. And again, that's through partly through the DPCC, the Data Processing and Coordination Centers. We envision that, we would like for that to be a good outward-facing, way of communication to the public, to be able to engage people more and I think one of the challenges we face is always, and this isn't just specific to the CEIRS, is making people aware of the resources that we have that can be used, especially those from outside of the field. So it's something, that we're working actively to do now.
As you know, the National Science Foundation, when they give these center grants, there is a communication component required of each grant. You have to have a proposal to do something, podcast, video, blogging, that engages the public.
Right.
But you don't have that for the CEIRS yet.
Not specifically. If you were, again, to look at our council presentation that just occurred a few weeks ago, we do talk a litlittle bit about the future plans in that area. Yeah.
Just saying, because Microbe TV is available for such work.
Yeah.
Because we have made proposals for NSF applications to, for example, make videos to highlight what individual labs are doing, to do podcasting and so forth. I think that's really important because the public is paying for this, right? It's tax dollars.
Right.
And they can't read journal papers. And so other things are needed like podcasts and videos and so forth. I think that would be a great thing to do. So what would you say are some of the major accomplishments of the CEIRS since its inception?
It's hard to say. There have been so many. I think more broadly, I would say that without citing specific publications, because there really are so many. Every year, the CEIRS produce, some of the highest impact publications in the influenza field. I think that happens on a regular basis. And historically, I think a lot of that has been a lot in the surveillance and basic virology area. And increasingly, we're seeing, as these cohorts are getting folded into the CEIRS, a lot of really fundamental understanding about the immune responses to people who are infected, following infection or vaccination. So I think they've made a lot of important contributions.
But there hasn't been, say, a vaccine outcome from CEIRS work yet, right?
Not yet, but I do think that they play a very important role in informing where we need to go. There have been a number of talks, especially over the past couple of days, about understanding what is happening in humans as they are. Flu is fairly unique. Not only are you infected multiple times throughout your lifetime, it may be that the first infection you have plays a bigger role in your subsequent immune responses, but you're also sequentially vaccinated. All of these things have, an important impact on, your immune status in this year. And so understanding that is really complicated. And so the work that the CEIRS is doing today to help understand that, as well as the interaction with the virus, which is constantly changing, I think is really critical to informing new vaccine approaches.
Are antivirals part of the program as well?
Antivirals are a smaller part of the program, but yeah, a lot of the fundamental, I would say, not so much in discovery of new antivirals, but certainly understanding antiviral resistance, and again, the basic discovery of potential new targets that could be, that you could design antivirals around, I think, but the basic virology and host virus interaction.
So this meeting is an annual event for the CEIRS centers?
It is,
that right?
yeah.
So they all get together and talk about their work?
Yeah, every year, and I would say it's in my short time being with the CEIRS, it's a really great meeting, a very interactive and collegial group. I think over time, they have really done a great job of building a group of people who like to work together and are willing to share data that may not be published yet. And so it makes for a really great environment.
And these meetings are at the different sites?
That's right.
They
every
rotate
year.
among them?
Right.
All right. Anything else you want to tell our listeners about CEIRS?
No, I mean, I think the CEIRS have been and will continue to be a great resource for the influenza field. And so, and I also think there's lots of opportunities for collaboration for those who may not currently be in the field.
All right, thank you. All right, Florian, why do we need universal flu vaccines?
Well, because the virus is mutating all the time, and we're always trying to catch it, but it's faster than we are with regular and current vaccines.
All right, so what is a universal? Give us a definition.
Okay, so we can have a very long discussion about that.
Well, you can't, so you have to...
So in my opinion, a universal influenza virus vaccine is a vaccine that covers all influenza A subtypes and influenza B viruses.
All A and B.
Yes.
And is there anything implied about durability, how long it will last?
Well, yes, I mean, that's actually one of the most crucial points about a universal influenza virus vaccine. If you would have a vaccine that is universally protective, but the protection only lasts for 6 months, for example, it would be of very limited use, right? It could be useful in a pandemic outbreak where you can just use an unspecific vaccine, a universal vaccine, and protect people against the virus that just emerged. But you want to have long-term protection. I mean, I think the goal in the end is to have something that you get, like a polio vaccine, maybe 2, maybe 3 times as a kid, and then you're protected lifelong against infection. I think that's the ultimate goal. The question is how long it will take to get there.
Yeah, lifelong is great because I think one of the issues with the current flu vaccines, from what I hear from, say, listeners, is they don't want to go every year and get another one.
Exactly, and that's one of the reasons probably why the coverage is lower than with other vaccines, because you have to do it every year and that's a hassle. But there's another component to that a lot of people don't think about, right? So it's really, really important to realize that flu vaccines are vaccines for rich countries. Low and middle income countries don't have the infrastructure and the money to get people revaccinated every year. But now if you would have a vaccine that you give like a polio vaccine, all of a sudden you can implement that into vaccination programs and you would make the vaccine available worldwide, right? And if that vaccine has broad protection, you would actually decrease the chance that a pandemic happens, and you would probably get a lot of vaccine coverage worldwide. And right now, if you look at low and middle income countries, flu vaccines are basically not used there or to a very small percentage.
I think I've heard Tony Fauci say even 10 years, once every 10 years would be better than the current situation, right? Right, so minimum 1 year, but longer. All right, so there are a number of approaches to universal vaccines, and your lab exploits one or several. So tell us how you're approaching this.
Well, just to point it out, this is a collaboration that we have at Sinai between Peter Balesi's lab, but Adolfo Sastri's lab and my lab. And so one of the prime targets for universal influenza virus vaccines is the stalk domain of hemagglutinin. Just for a lay audience, the hemagglutinin is the major surface glycoprotein of the virus. It's the part on the surface of the virus that makes the virus stick to the cell. So without it, it can't get into cells, right? And it's kind of looking like a mushroom. And you have a cap, or the head of the hemagglutinin, which typically vaccines induce antibodies to, and they're super neutralizing, but this head or cap changes all the time, right? And then you have a stalk, and that stalk is relatively conserved. It usually doesn't change that much. And so if you have antibodies against that stalk, they are actually cross-reacting and cross-neutralizing different subtypes. So it can go from H1 to H5, for example. The problem is that the immune system is not very fond of that stalk domain, and you don't get a lot of antibody responses against it. And so what we're trying to do is to trick the immune system to make more of these antibodies.
Do we understand why the immune system doesn't recognize the stalk as much as the head?
Well, there are a number of hypotheses. I don't think we're completely clear about it. One might be accessibility. So a soluble antibody can easily fit in there and neutralize a virus by binding to the stalk. But if you think about B cell receptors, right? So I always imagine a B cell like, it's covered by basically trees, right? And some of these trees are B cell receptors. And then you have the surface of the virus, which is covered by hemagglutinins, and it's also pretty dense, right? So I think the B cell receptor has a hard time to actually find the stalk. And that might be one of the reasons why we don't get a lot of these antibodies. But there might be other reasons involved too. There is more and more studies into that, but I don't think we have a really good idea about it.
As Brianne Barker on TWiV recently said, the immune system goes for the shiny parts. That doesn't tell you how it works, though. Now, in people infected with influenza virus or vaccinated, do they make antibodies against the stalk?
Well, there's a huge difference between what happens after vaccination and what happens after infection. And so after infection, you can get some levels of these antibodies. And they seem to be part of the natural protection that we get after we got infected. After vaccination, not so much. So there's extensive studies that looked at that, and there's basically very little induction of these antibodies after regular influenza virus vaccination.
But after infection, there is. But you said earlier it's pretty rare, right?
Yeah, I mean, you see these antibodies, they don't go to extremely high levels, but we just did a study together with Aubrey Gordon where we looked at the levels of these antibodies induced by natural infection and then how they protect against subsequent H1N1 infection. And it turned out that they, even at lower levels, are an independent correlate of protection. So, I mean, I think the key is for flu vaccines in general, the key is to understand what infection does, what vaccines don't do, right? Because the other, and you talked about longevity of antibody responses, right? Influenza virus infections induce very long-lived antibody responses. I think John Drainer did a study within the CEIRS network a few years ago where they looked at H2 antibodies in the human population, and H2, stopped circulating in 1968. And people still have sky-high antibody titers against it, and that's from natural infection. But with vaccines, you have waning sometimes during the season. Somebody told me that Stanley Plotkin is getting flu vaccines twice, once in the beginning of the season and then in the middle of the season to make sure that he's really protected, right? So I think understanding these differences would really help us to come up with better vaccines.
So what is your approach to tricking the immune system to see the stalk?
So from exposure to natural infection and to vaccines, we all have memory B cells that have specificities for the stalk domain. So you have memory responses. You just don't have very high antibody levels. And so the idea is to vaccinate people with what we call chimeric hemagglutinins, which are combinations of exotic head domains from avian influenza subtypes, and then with H1, H3, or influenza B stalk domains. And so if you vaccinate somebody with a construct like that, the immune system hasn't seen the head before. So there's, if any, a primary response against the head. But the immune system has seen the stalk, and so you get a recall response. And the idea is to use these constructs sequentially, always changing the head, but keeping the stalk constant. And then by doing that, boosting antibodies against the stalk. So
does this work in animals anyway? You've tested it, right?
So in animals, it works pretty well, and you can protect against a wide range of different subtypes. And so we have clinical studies ongoing, phase 1 studies, that also look at that, and we got interim results, and there's limited data and no data on longevity, but from the data that we have, it looks relatively good.
So in the animal studies, you get protection against challenge, correct?
Correct.
And do you know anything about the durability? Is there memory? I know the animals don't live a long time, right?
Well, mice lie, and ferrets are kind of not really honest either. So if it would be about animals, I mean, in mice we had titers that were stable for a year, which is basically the lifetime of a mouse. In ferrets, we also had stable titers for a year, and then when the animals were challenged after a year, they were still protected. But I think one of the big challenges for vaccine development, not only universal vaccines, in general flu vaccines, is that, you have an animal model. Animals are relatively naive. Even if you prime them once or twice with natural infection, you don't get to induce these complex memory responses that humans have and all this pre-existing immunity. So I think the only real animal model that exists for humans is humans. And so you can do amazing things in animals, but that doesn't mean that they translate into.
Right, so you're in phase 1 at the moment, it's ongoing?
Yeah.
It's basically a safety study, right?
Yeah, but you can do, obviously the primary endpoint is safety to look if it's safe to do it. But we do a lot of immunogenicity. There's a lot of assays that we're performing. For most of it, we don't have access to the data yet because it's blinded. But we have some data for the interim results. So it's a very interesting study.
Okay, and then presumably you'll go to a phase 2, which will involve some indication of protection, right?
Well, yes, we are not sure yet how we're going to do that. One idea would be to do a human challenge model to kind of look at protection right away. I think BARDA did that recently with Vaxart, with an adenovirus vaccine, and that seemed to work really well. So that would be a possibility. But I mean, the general question with universal flu vaccines is, and we have people from the FDA here who might be better qualified to talk about that, but the question is, how do you license it, right? Because if you want to claim universality, you have to prove that in a phase 3 study, right? And so how do you prove that? And I think that's going to be tough.
You can never challenge against all the known HAs, right?
Exactly. So the question is, how is that going to be viewed from a regulatory perspective, and what's then going to be really on the label of your vaccine, right?
Well, I have a feeling it's not going to be universal.
I have the same feeling.
But if you restrict it and then it works beyond that, then you can add that, right?
Yeah, but I mean, that's a whole different question. I mean, it takes time to go to a phase 3 study.
Are you also doing similar things with influenza B viruses?
Yes. So the idea that we have, these stalk-reactive antibodies are usually broad, and some are exceptionally broad. They can go from A to B, but typically they're broad within a group. So there's group 1 influenza A hemagglutinins, group 2 influenza A hemagglutinins, and then B. And so our idea is that we need a trivalent vaccine with a component that covers group 1, a component that covers group 2, and a component that covers B. But honestly, I think B is the easier target because you have less antigenic diversity.
Mm-hmm. You have basically 2 antigenic types circulating, right? Lineages, correct. So that's all you have to cover.
Correct, and they're closer than the 2 influenza A subtypes, for example.
So there, you could call it a universal B vaccine, right?
Yeah, I mean, I'm always, Arnold Monto is here, he's always telling me that I'm crazy, but so influenza A has an animal reservoir, right? Influenza B does not. Yeah. And so hypothetically, you could eradicate influenza B with a good vaccine and a high vaccine coverage. And I mean, I'm kind of dreaming about that. It's not very realistic, but it's a possibility.
I just came from a meeting where there was a lot of discussion of phage therapy. And it's quite clear that FDA is going to have to change how it licenses things to license a phage to treat bacterial infections. Because, for example, they'll have to be tailored to the individual infection. So it's not going to be the same as a drug, which is one size fits all, right? It's quite interesting. Well, there's so much more we can talk about. I think, Florian, I have to come to Mount Sinai, or you come to my office and we can dedicate an entire episode to talking about this. But what is your outlook? Are you optimistic that we can get such a vaccine that will cover a lot of strains and be durable? And if so, what's the time frame? A year, 5 years, 10, 50, or 100?
So I'm a very negative person, but I think we can do it. I can tell you about the time frame. So there's 2 different things, right? There's ways to improve the current vaccines. There's now a huge focus on neuraminidase, and I think we can get the coverage and the vaccine effectiveness up with having better neuraminidase preparations in the vaccines. And there might be other ways to actually make the seasonal vaccines broader within a subtype. But I think the ultimate goal should be to have a universal influenza virus vaccine. And we're focusing on this dog, but there's other targets too. I mean, one of currently my favorite influenza virus protein is actually neuraminidase, and I think there's a lot of potential. But people are also looking at antibodies that bind to the head domain, to conserved sites in the head domain, which is also a challenge. And then obviously there's M2, which has been a target for universal flu vaccines for a long time. And you can also get a lot of benefits from T-cell responses to internal proteins, which are relatively conserved. So, in the end, we might have a vaccine that is a combination of these mechanisms and these targets, right? But I think it needs to be tested. What we really need is more human trials to actually figure out what immune responses we get. And so I think that's really the important part, to figure out what happens in humans. I don't think we can learn too much in respect of universal flu vaccines from animal models. But I'm very confident that we'll get there.
So 3 papers just came out, I think, describing these head domains, epitopes that are hidden, right? And are only exposed when the HA breeds, and they seem to be conserved, right?
Yes. And those are super interesting. I think George had the first antibody kind of that showed that, I think it was published in 2016. The interesting part there is that it depends on the stability of the hemagglutinin, right? So they're very good against H1 because the trimer seems to be loose and breathes a lot. Against H7, for example, which has a very stable hemagglutinin, not so much. But that might be an additional component that is helpful.
Okay. I want to make a plug for this terrific review article that you just published, The Human Antibody Response to Influenza A virus Infection and Vaccination, published in Nature Reviews of Immunology just this month, June 2019. So it's excellent, awesome illustrations. And you promised me you'll come to the TWiV studio and we'll talk more, because there's a lot more that we haven't covered.
Yeah, that would be terrific. And it's just a cap right away, right?
I'll pay for it.
Great, thanks.
That was it, right? Just me paying for it. All right, let's end up with Jennifer. You're interested in cellular responses.
Yeah, I've been interested in cellular responses since I started in influenza. More with a focus on pediatrics recently and how immunity develops in children. So right now, really what my study is looking at a cohort of children between the ages of currently 3 months and 15 years old, and looking to see how immunity in general, but my particular focus is on CD4 T cell responses develop and how that's different following a first vaccination, an early infection, and at what point do these children start to resemble adults. So this is going to be really important when you're talking about universal influenza vaccine development. It's known that kids can have much more durable responses to influenza than adults do. I mean, this has been known since 50s and 60s with, original antigenic sin has found new life in the idea of imprinting. So what's special about responses in children that allows them to be sustained and actually be recalled upon, vaccination as a young adult, or even in some cases an older adult? And if we could understand that, then there is potential to try to elicit that kind of response in a vaccine that can be administered to a more naive child. I'm not going to say necessarily totally naive, but a children who's had fewer influenza exposures. And you could potentially get that kind of durable reactivity with a vaccine as opposed to against a single strain with infection.
I've been waiting for my kids to resemble adults for many years now.
Yeah, me too. My oldest is 12 though, so.
When do kids first get a flu infection typically?
So I think it's highly variable. It depends a little bit on where you are, the kind of living situation you have. We know that in areas with high crowding, you'll get an influenza infection much younger. I think it also depends to some extent on vaccine. The data I have from the kids who I've enrolled thus far suggests that kids who are, even 3 and younger have very, very different reactivity than what older kids and adults do. And one of the things that I'm interested in is trying to find, a marker to say, okay, this kid's been infected versus a kid who hasn't. we are very good at tracing vaccine histories in children because in kids, vaccines are reported to often databases, for instance, NICES. Infections are much harder to track. I've been, reaching out to families, are kids feeling any illnesses, but, kids can be sick, their entire first year in daycare sometimes. So it's hard to know that. So if we could have a marker for when a kid's been infected, that would be great.
And when do we typically immunize kids? At what age?
The recommendations in the United States are for immunization starting at 6 months. So that's another area of interest in that when we look at older data, when you look at adults now and say, oh, look, this immunity from infection has persisted, those adults probably weren't immunized and probably had their first influenza exposure through an infection. Whereas now we are, in many cases, actually immunizing kids before they become infected.
And what coverage do we have at that age? Do you have any idea?
It's highly variable depending on where you are. Different communities have very different vaccination rates. Rochester in general has a relatively high vaccination rate, but it's variable.
I bet it's really low in Texas.
And
where overall vaccination is pretty low.
In areas where overall vaccination is low, influenza vaccination is low. And I think influenza vaccine faces an additional challenge, especially as, you get numbers put out, about the poor effectiveness of influenza vaccine. We have, our population has been great in terms of involving their kids in research. We have an amazing set of kids, an amazing set of parents, but one of the most common questions that I'll actually get is, well, why should I immunize my child for influenza? I just saw on the news or the radio or whatever that, vaccine effectiveness was, 20% this year. And one of the things I like to do is, from the clinical side, is to point out that yes, your kid may still get influenza, but it might prevent, the vaccine very well may prevent them from getting severe disease and ending up hospitalized.
And to which they say you're just a pharmacy shill wanting to sell more vaccines, right?
Actually, I haven't gotten that one very much, but obviously the people who are coming to participate in an influenza vaccination study are generally not hardcore against vaccines.
I have on the podcast, I recommended people say, you get money from the pharma companies. That's why you say that. I wish I did get money from them. I don't.
But if we can understand why immunity in children is especially durable, then that could be harnessed to design a vaccine that could produce such durable immunity, into adulthood potentially.
So you've published a couple of papers on CD4 T lymphocytes, right? So let's talk a little bit about what they are and what they do in an influenza virus infection.
So when you're looking at the CD4 T cell response to influenza, the easiest thing to talk about is, well, they provide help to the B cells. And that certainly is a very important thing. So you'll have your CD4 T cells, specifically T follicular helper cells, that are able to go to the B cell zone of the lymph node, that are able to provide help so that you get a affinity maturation of your antibody response and you get a better antibody response. However, I agree with what Florian said, that when you have a vaccine, if you could harness multiple arms of the immune system, it's going to be a much more powerful vaccine. The hard thing about CD4 T cells is they have a lot of functions in influenza infection. It's not only B cell help. You also have help to promote an innate immune response. You actually have CD4 T cells that can be cytolytic and have been shown to be cytolytic in influenza infection. And understanding the balance of that function and what is going to be protective versus what's going to be neutral versus what's going to be harmful, I think is a real challenge. Andrea once put up a slide that had the CD4 T cells depicted as smiley faces or angry faces. And really, there's a lot of work that still needs to be done to understand what is the best kind of CD4 T cell response to elicit.
So you published a study where you said, so the way the CD4s work is they have a receptor that will recognize a peptide presented to them by antigen presenting cells.
Yeah.
You did a study where you asked of all the possible peptides encoded in the flu genome, which are recognized by CD4s from infected kids, right? You compared young and older kids.
So I looked, in that particular study, what I did was I looked not at the epitope level, because the cells are present at relatively low frequencies that would be a challenge to find, but I was more looking at the protein level. And I compared younger children, so these were all kids who were younger than 3, and I looked at what their CD4 T cell reactivity looked like, so what's the specificity of their response, and I compared that to data that we had available on the adult population. I found that there were a lot of differences in the CD4 T cell reactivity between young children and the adult population. One of the most remarkable was CD4 T cells against NP and M1. These are more, I won't say conserved, because we know that these proteins undergo, mutate as well, but they're much more conserved than, say, the hemagglutinin is. And what I found was that young children have very little reactivity to these internal virion proteins, likely because many of them haven't been infected yet. So their exposures have been predominantly through an inactivated vaccination that contains very low amounts of the NP and M1 proteins. Now, in adults, when you're giving a pandemic vaccine, so a vaccine against which you don't have pre-existing memory to the hemagglutinin protein, you get boosting of these NP-specific CD4 T cells because you preferentially boost memory responses. I would hypothesize, at least, that there's not enough NP in the inactivated vaccines to establish that kind of immunity when you're talking about a naive CD4 T cell response.
So either we need to use an infectious vaccine or supplement?
So the novel LAIVs, I think, are actually potentially really interesting. I know there's some work going on looking at using the chimeric HAs on live attenuated vaccines, because they'll get immunity to the right place. So you'll get immunity within your respiratory tract. They're better at stimulating CD4 T cells and CD8 T cell responses. So I think that's a really interesting route. And I know there is some earlier, like phase 1 studies looking at kind of novel live attenuated influenza vaccines. And actually, I believe it's a pediatric population, like 9 to 17 years old. So.
Would it be interesting to do a study of children only immunized with LAIV and look at the CD4 response as you did in that other study?
Yeah, that's something I'm really interested in doing. One of the challenges that we face right now with our current LAIVs is they're only recommended starting at the age of 2, and current vaccination recommendations are starting at 6 months. Now, there's a lot of kids who aren't vaccinated starting at 6 months, so I think we could find the population, but we couldn't, given what current recommendations are, and that influenza vaccines can prevent infections and even especially severe illnesses, we couldn't say don't get vaccinated.
Right. So earlier you published another study which suggests you could prime CD4 cells to overcome this bias. Mm-hmm. Can you briefly summarize that for us?
So I think what you're referencing is a study that I actually did as a fellow in Andrea's lab. And we were looking at a pandemic H5 vaccination. So this was a population of people who previously had been vaccinated against H5N1 influenza as part of a study that was done at University of Rochester. And what we found was that when you had administered an inactivated H5N1 vaccine and established CD4 T cells, especially CD4 T cells against the HA protein, you were able to get boosting of the antibody response upon vaccination the following year. So it was actually, I think it points to the fact that CD4 T cells, establishing the right memory. Now, we don't know what the phenotype of those cells were. All we know is that they were there, but presumably some of them were follicular helper cells that were able to go to the lymph node. If you're able to establish that population of cells, you can potentially have improved antibody responses even against, potential, pandemic or potentially pandemic influenza strains.
All right. Well, I have to stop, unfortunately. We could go on for many more hours, but the next session is going to begin. So let me end by just saying that TWiV you can find on podcast players. If you listen, please subscribe. We can know who's listening. That helps us to raise money. And you can do that on any podcast player. We love questions and comments, twiv@microbe.tv. And if you like what we do, consider supporting us financially, microbe.tv/contribute. You could give us $1 a month. That would really help because we have many thousands of you listening. My guest today from NIAID, Alan Embry. Thank you so much.
Thank you.
From Icahn School of Medicine, Mount Sinai, Florian Kramer. Thank you very much.
Thanks.
Come back. And from the University of Rochester School of Medicine, Jennifer Nyack.
Thank you. It's been a unique experience.
I'm Vincent Raccagnano. You can find me at virology.ws. I want to thank Andy Pekos and the organizers of this meeting for inviting TWiV and ASV and ASM for their support, and Ronald Jenkins for his music. And before I wrap up, let me give you some t-shirts. Who's a TWiV fan? I don't know if I can reach you, but I will try. You have to walk for it. Who else is a TWiV fan? Right here. Who else? I have two more. Who else is a TWiV fan? I don't know if I can reach you, but I will try. One more. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.